The science and applications of synthetic and systems biology. Humans have been modifying the genetic characteristics of plants and animals for millennia by controlling the breeding of species in order to select for certain traits or characteristics and to reduce or eliminate others. The discovery of the structure of DNA in 1953, the recognition of its importance as the carrier of heritable genetic information, followed by the development of recombinant DNA technology 2 decades later, paved the way for powerful technologies to manipulate genes directly and in such a way that the genotype and phenotype of an organism can be altered with utmost precision in a single generation. The realization of viral and microbial genomics in the last few decades of the 20th century, coupled with the completion of the initial draft of the Human Genome Sequence in 2001, reflect a fundamental shift in the way biology is studied and has opened a portal to vast post-genomic possibilities. Because of the Human Genome Project, scientists have already identified more than 1,800 genes associated with particular diseases. More recently, scientists have developed techniques to more effectively synthesize or modify larger segments of DNA, making a significant change in the way people study biological systems and a growing capacity for both experts and amateurs to manipulate such systems. Until the past decade, the work was often painstakingly slow and able to address only relatively straightforward challenges, such as the manipulation of one gene at a time. More novel or complex genetic modifications would be difficult to construct using the more conventional recombinant DNA, rDNA techniques of the 1970s and 1980s. In the past decade or two, newer approaches combining engineering and biological techniques have enhanced researchers' abilities to manipulate DNA. These new synthetic techniques allow for genes and long chains of DNA to be designed and constructed from scratch using a computer and relevant chemical compounds, rather than employing a trial and error approach to the identification and insertion of pieces of existing genes from living cells into a novel host environment. In May 2010, researchers at the J. Craig Venter Institute announced that they had produced the first functional, self-replicating bacterium whose entire nuclear genome had been synthesized artificially in the laboratory, albeit using a naturally occurring genome sequence as a template. While the achievement did not, as some media reports at the time suggested, represent the creation of life, it did propel the nascent field of synthetic biology into the mainstream and generated a number of questions and much speculation about the potential power, utility, and risks associated with work in this field. Although biologists may have a long way to go before they have enough knowledge and tools necessary to design and build life, the emerging field of synthetic biology has already produced several novel products and lead compounds for drugs and vaccines, fuel, biofabrication of materials, and other industrial applications. Most, if not all of these products and compounds are being generated via the type of top-down approach, with scientists re-engineering existing cells to do things that they do not normally do. By inserting the genetic machinery for metabolical pathways into Escherichia coli and other host organisms, scientists are attempting to create microbial biofactories for the production of pharmaceutical ingredients, flavors, fragrance, and other chemical products. The goals also include compounds in cells with new phenotypes and functionalities, such as cells that can produce carbon-neutral biological fuels with properties that are similar to those of petroleum-based fuels and novel drugs. The United Kingdom's Royal Academy of Engineering observed that systems biology aims to study natural biological systems as a whole, often with a biomedical focus, and uses simulation and modeling tools in comparison with experimental information. Synthetic biology aims to build novel and artificial biological parts, devices, and systems. Many of the same methods are used, and as such, there is a close relationship between synthetic biology and systems biology. But in synthetic biology, the methods are used as the basis for engineering applications. While both disciplines use similar approaches, systems biology uses these approaches to better understand the inner workings of life, whereas synthetic biology emphasizes the application of the lesson learned from systems biology for the purpose of engineering, or re-engineering, living systems to behave in specified ways. Many potential applications of synthetic and systems biology are relevant to the challenges associated with the detection, surveillance, and responses to emerging and re-emerging infectious diseases. On March 14 and 15, 2011, the Institute of Medicine's Forum on Microbial Threats convened a public workshop in Washington, D.C. to explore the current state of the science in synthetic biology, including its dependency on systems biology, discuss the different approaches that scientists are taking to engineer or re-engineer biological systems, and discuss how the tools and approaches of synthetic and systems biology were being applied to mitigate the risks associated with emerging infectious diseases. Through inventive presentations and discussions, participants explored the ways in which synthetic and systems biology are contributing to drug discovery, development, and production, vaccine design and development, and infectious disease detection and diagnostics. In addition, workshop participants considered how synthetic biology could be used to engineer or re-engineer microbial host cells to detect environmental toxins, produce carbon-neutral fuels, and produce novel raw materials. So now, let's hop on over here to the MIT. Synthetic biology circuits can respond within seconds. MIT engineers designed the first synthetic circuit that consists entirely of fast, reversible protein-to-protein interactions, and this was released on July 1st, 2021, so it's over 4 years old. Synthetic biology offers a way to engineer cells to perform novel functions, such as glowing with fluorescent light when they detect certain chemical. Usually this is done by altering cells so they express genes that can be triggered by a certain input. However, there is often a lag time between an event such as detecting a molecule and the resulting output, because of the time required for cells to transcribe and translate the necessary genes. MIT synthetic biologists have now developed an alternative approach to designing such circuits, which relies exclusively on vast, reversible protein-to-protein interactions. This means there is no waiting for genes to be transcribed or translated into proteins, so circuits can be turned on much faster, within seconds. If we scroll up to the image, this is a synthetic biology circuit. This is how they create synthetic biology.